the case where ontologies are used to facilitate consistent description in databases across organisms. And so that's the motivation, as we know, for the gene ontology. And I just put a snippet of the gene ontology up here so you can get a sense, for those of you who might not be familiar with it, of the sorts of terms that are in the gene ontology. The gene ontology itself is probably one of the most successful biological ontologies in existence. And just some basic statistics on it so that you understand the sorts of the scale that we're dealing with. So there, as of June 9th, there were 27,602 terms split across 3 different branches, as you see there. There are currently annotations into the gene ontology for over 40 organisms. I did a quick search in PubMed to see how widely gene ontology was mentioned in PubMed abstracts, and I found 2,068 publications, and I did a similar search in the ISI Web of Knowledge, and there are over close to 4,000 references to the primary gene ontology paper. So this gives a sense of how widely it is used, and I think it gives an indication of how important it is to have it be a high quality resource. It's really in widespread use, and yeah, so it should be of high quality. Now, in fact, it turns out that it is, and so I will tell you some numbers later, which I think you might find interesting. But one of the key quality concerns that was raised at the meeting that we attended was what is called univocality, and in fact, that term goes back really far in philosophy. And it was originally coined to mean a shared interpretation of the nature of reality. And David Hill from the Jackson Laboratory actually revived it at this meeting and intended it to mean a slightly different thing, but very closely related to the original usage, which is consistency of expression of concepts within the ontology. And it turns out that the gene ontology is, in fact, highly regular in its expression in the terms. And this is a very good thing because it facilitates human usability. But I think more importantly, it allows computational tools to utilize the regularity of the structures. So for things like alignment that we heard about in the last talk, having a consistent form of expression of these terms means that it's more easy to align things together. So the sort of variation that one might expect to see in a large resource like the gene ontology are the examples at the bottom, where on the left-hand side we have terms that are actual gene ontology terms, so regulation of transcription and positive regulation of cell migration. And on the right-hand side, we have terms which are basically syntactic variants of those terms, but are not gene ontology terms. So regulation of transcription can be expressed as transcription regulation, and positive regulation of cell migration could be expressed as cell migration positive regulation. And however, there's a sort of a predominant form that's used in the gene ontology, and in order to have consistency, we would like to avoid having variations of the sort we see on the right. So our goal was, in fact, to try to identify cases in which there were violations of univocality, so in which there were cases where two similar, semantically related terms were expressed with a different structure. So the problem here is essentially that the Go is generally of very high quality. And so how do you go about trying to find these cases of univocality violations, these inconsistencies, when you have over 27,000 terms? So if you start going through it pairwise, you're going to have over 700 million combinations to look at. That's not a fun job. I don't think anybody wants to do that, unless there's some volunteers in the audience. And so, in fact, we need some computational tools to help us work through that. The hypothesis that we were working from was that violations of univocality would correspond to transformational variants of the term structures. And we decided to take a very simplistic approach to this. So you'll see that the strategies that I describe in a minute are really very simple. And the reason for that is, Occam's razor, try the simplest thing first. We wanted to see if by doing a very sort of shallow analysis of the terms, we could identify these univocality violations. And this also improves the generalizability of the algorithm to other ontologies. And we'll see we get quite far, although there are some problems, and that suggests that some more linguistic analysis of the terms might be helpful. So the strategy that we have taken in this work is term transformation and clustering. We do three transformations, the most important of which is abstraction. And what abstraction corresponds to is the substitution of embedded gene ontology and CEBI terms. CEBI, excuse me, is another ontology. It stands for chemical entities of biological interest, and they are chemical entities. And the gene ontology terms often reference particular chemical entities. And so what we do is we simply look for gene ontology terms which contain terms from either the gene ontology itself or the CEBI and abstract them with a variable representation. So any gene ontology term is abstracted into a G term label, and any CEBI term is abstracted into a C term label. So we see here that we have toluene oxidation via 3-hydroxytoluene, and toluene and 3-hydroxytoluene are both CEBI terms. And so that phrase with our abstraction transformation gets abstracted to C term oxidation via C term. Similarly, with regulation of coagulation, we have an embedded gene ontology term there. So this stands on its own as a gene ontology term inside of this larger gene ontology term. So we abstract this to regulation of G term. In the third case, we have an example where there are both within one gene ontology term. So leukotriene is a CEBI term, and acute inflammatory response is a gene ontology term. So this phrase gets abstracted to C term production during G term. The second transformation that we performed was stopword removal. And stopwords is an information retrieval concept which basically says words which carry very little semantics are basically there for, if you will, syntactic sugar to indicate the structure of a phrase. And so there are high-frequency words in English, words like the and of and via. And so we simply had a list of those, and we went through all the terms and removed them. And so what that does is, if you have a phrase like toluene oxidation via 3-hydroxytoluene, you just remove the via, and you end up with that phrase there. Similarly, for regulation of coagulation, you remove of and end up with regulation coagulation. Then the third transformation we performed was to do alphabetic reordering. And this was basically to deal with word order variation in the terms. And so we can apply these transformations in combination, and we tried all combinations. So abstraction alone, stop removal alone, reordering alone, and I'll show you some data later about the impact of each one of these things. But basically the idea, if we apply all three of them together, is that we take each of these terms, and the first phrase becomes C term oxidation, the second phrase becomes G term regulation, and the third term becomes C term G term production. Now, when we apply all of these transformations, we end up with a sort of canonical pattern for each term. And to do clustering, all we did was simply look for common canonical patterns across a set of terms. So we're grouping together all of the terms that have a common form after the transformation step or steps have been applied. And as I said, we did this for different combinations of transformation. So as an example, if we apply all three of the transformations, to these sets of this set of terms, we end up with the pattern G term structure. So what we've done here is also a bit of stemming, so we've got rid of the endings of words, and that's why this doesn't look like actual words. But this also helps us deal with a bit of the variation in morphology or inflection that you see in the words. So all of these forms, structural constituent of cell wall, structural constituent of chorion, and so forth, collapse to this basic structure, G-term constituent structure. And we see here that we've found one term which is inconsistent with the rest of the terms here. So extracellular matrix structural consistent has a different syntactic structure than the remainder of these terms, and so we would say that this constitutes a violation of univocality. So it's an inconsistency in the expression. One might expect that this should be rephrased as structural constituent of extracellular matrix in order to be consistent with the other terms. So as I said, the abstraction transformation had by far the most impact on the set of terms. So if we recall that there were, so we worked with an older version of the gene ontology in which there were a little over 25,500 terms. And after doing the stemming pre-processing, that reduced to a set of a little over 23,000. That's because you have variants that have a chemical name with a 1 on the end or a 2 on the end and so forth, and we stripped those away. So that already gave us an 8% reduction in the number of terms we were dealing with, and or in fact that led to this many clusters. And then, so if we look on the left here, these are all of the transformations that had no abstraction in them. And these are the transformations where abstraction was done. And so here you can see, simply after the pre-processing, we had 23,478 clusters, where basically most of them just had a single term in them. There was at least one cluster that had 29 terms in it, and again, it was because of very, very minor variations of the term name, of words inside of the term. And once we apply the abstraction step, the number of clusters is basically halved. So abstraction alone reduces the number of clusters from 23,000 to 12,000. And we end up with one very large cluster, which has almost 3,000 elements in it. And so, clearly the abstraction step has the most impact. All of these, even after we've applied the abstraction, the reordering, stopword removal, or all of together only has a very minor impact on the number of clusters. But a lot of the univocality violations we found actually came into play at this level. So they were small changes, but they were significant. So if we look at abstraction more carefully, and we just see how much of the abstraction was due to embedded Kebi terms and how much was due to embedded Go terms, we see this breakdown here. And so you can see that both abstractions were actually important for achieving the clustering effect. And so, so if you look at the distribution of the cluster size before the transformations were applied and after the transformations were applied, note that the scales here are completely different. So, as I said, the maximum cluster before transformation had 29 elements in it. And after transformation, we had one that had 3,000. But basically, we have a number of very large clusters. So the abstraction is really making a big difference. Okay, so to jump to the punchline, we ended up with 237 candidate clusters that we thought may contain a univocality violation. And we identified those by what I call a heuristic search. So basically, we're motivated by the observation that the abstraction has the most impact, and that the other transformations cause certain things to collapse together because of the word order variation. And it's precisely the word order variation that we're interested in. So what we did was we looked for terms that were in different clusters after just the abstraction transformation step, but merge together into a single cluster, collapse to a common form after application of one of the other transformations. And when we did that, we ended up, as I said, with 237 clusters. Now, some of these were identical because applying both of two different transformations often didn't result in any difference in the canonical form. So alphabetic reordering may have no impact because the words may already be in alphabetical order. So once we remove those, we end up with 190 clusters that needed to be manually assessed and evaluated. And so, again, compare that to the 27,000 terms we started off with and the 700 million pairwise combinations of those. So we have really reduced the number of things that need to be looked at by hand, very much. So I did the analysis, went through them all by hand, and we ended up with 67 true positive cases of univocality violations, which have been submitted to the gene ontology curators, and they're working through them now. And 123 false positive cases, and we have some ideas for, in the future work, how to lower that number a bit so that the person who's going through this manually has even less work to do. So some analysis of the clusters that we have. So as I said, we ended up with 67 true positive clusters. This constitutes 317 Go terms. Now, we worked with an older version of the Go, so after applying an obsolete term filter, so looking for terms that are now obsolete in the gene ontology, we removed 7 of those clusters, which contain 32 terms altogether. And based on my analysis, I anticipate that there'll be on the order of 75 or so rephrasings that will need to be done in the gene ontology for those 67 clusters. So the kind of thing that we found is here. So one very common thing is to have a transformation of a phrase like XY into Y of X or Y in X. And this was about 45% of the cases of true positives that I identified. So an example of this is here. So if you have induction by organism of symbiont apoptosis and you have induction by organism of systematic acquired resistance in symbiont, you see that we have 2 terms here that really, to be consistent with the other 4 terms, should be phrased, this one should be phrased as induction by organism of symbiont systematic acquired resistance, and similarly for the second one. So by doing all of our transformations, we're able to identify that sort of violation of univocality. A similar example is down here at the bottom. If you have multicellular organism reproduction and reproductive cellular process in multicellular organism, this suggests that there's some variation in phrasing that should be addressed. It's not up to me to decide which is the correct one, the canonical form, and which is the incorrect one. It's really up to the curators. But in any case, we've identified a potential case that might need to be addressed. So then there were many cases where there were determiners in one form and not in the others. So here we have in the forebrain, and here we have in forebrain. So this is a very small difference that is easy to fix. So other examples of true positives are here. Some of them get down into the noise, but commas here carry some semantic meaning, sometimes with a preposition, sometimes without, and these are variations that are important to address. So then there are some smaller things, again, within versus in, just words, very specific things that we identified. The false positives break down like this. I'm running out of time, so I'll go through very quickly. But by far the most important reason for a false positive in this data set was that some of these stop words that we remove actually carry meaning. So in this case, the bys and the ofs tell us the roles that the various elements are playing. So bihost of symbiont RNA levels and bisymbiont of host RNA levels actually express as a different semantics. And so by eliminating those stop words, we've actually removed the important difference between those two phrases. And then similarly, the difference between at and in is actually important semantically. Some of them we just ended up with, by doing so much abstraction, that we just had completely different structures. And I will jump ahead. And so basically the conclusion is that we use simple term transformations and a heuristic search to identify 67 true positive clusters in our data set of potential univocality violations. And we have some ideas for doing more specific analysis of the linguistic alternations. And Kevin Cohen, my colleague, will have a paper on normalizations later in the week, which will address how some of that actually manifests itself in biological text. And so we will build on that to try to improve the true positive rate of our analysis. Thank you.
Okay, do we have any questions? If not, then I'll ask one. So it looks like gene ontology is a beautifully clean ontology.
Right.
Have you thought about applying this to something that's maybe a bit more rough and ready? And who would you choose first?
I shall not name names. No, yes, certainly we've thought about applying this to something which is less clean. Of course, for something which is less clean, it's a lot easier to find the univocality violations. So it may be the case that the clusters end up so huge that that's even too much. So then we may have to do some more specific linguistic transformations or whatever to try to make those kind of more narrow. But yeah, certainly this is the tools will be available probably this week on our website, and they can be applied to any OBO ontology. So if anybody wants to try it, feel free. And yeah, we will definitely try it on other ontologies.
Any more questions?
You're welcome, Judy.